Hello, I'm Dr. Francine Marques from Monash University, and I'm here with the two winners from the Hypertension Council Awards from the basic and the clinical science. So Dr. Giacomo Rositto and Karen Clark. Welcome.
Thank you very much.
Giacomo, can you tell me a little bit about your study?
Sure. So what I try to present as part of my work on new concepts related to how salt associates with cardiovascular disease. And in particular, there are some novel concepts which would suggest how salt can induce cardiovascular disease independent of blood pressure via changes in metabolism. And that's what we explored in a quite unique population of patients, a very carefully characterized group of patients in the University of Padua, which were our collaborators. And in this cohort gave us the chance to show how our kidneys are able to handle in different way salt and water upon high salt intake, which is a condition which would otherwise lead to some loss of fluid and fluid imbalance, to a point that the more salt you eat, the more your kidney is able to deal with this salt, preserve water, but obviously spend energy to this extent. And this, in the end, would result in changes in metabolism, a sort of catabolic state, which would induce a loss of important proteins, and overall this shift, which can be in the long term detrimental in terms of cardiovascular risk.
Okay, amazing. So can you tell me in one sentence, what is your take-home message?
My take-home message is, in a sense, the more salt you eat, the more you would eat.
Okay, yeah, amazing, yeah. And Karen, you are doing some studies looking into a genetic model that lion rat, is that right? Yeah. Can you tell me a little bit about it?
Sure. So our lab's mission is using genetics and genetic tools to discover new ways to understand hypertension and obesity and dyslipidemia, anything that's involved in the metabolic syndrome. And the really cool talk that we had on the first day of the 1,200 new SNPs that they found and validated is really interesting. But the thing is, like, I agreed with one speaker who said that that's way too many to look at, and that's for a couple of reasons. A, nobody's going to have all 1,200 risk loci. That would just be astronomically horrible, and they would have the worst luck of all time. But, the thing that's important is, like, finding things that are in novel pathways that you may not have expected. A lot of people say, like, oh, we didn't expect this gene because it wasn't in renin or something. And, like, there's a lot of genetic mechanisms that contribute in ways that people don't expect. So we had a rat model where these two rats only have maybe 1,000 non-synonymous variants, but it's like the difference between a runway model and a sumo wrestler in terms of phenotypic diversity. So clearly there's not a lot going on that's different there genetically, and yet phenotypically they're amazingly diverse. So we decided that that was a really excellent model to untangle the genetic causes of disease. And so we had classical QTL mapping, we did eQTL mapping, and the areas that we focused on were important in both studies. And then within that area, there's not a lot of variation that we had to go look at because these two strains are so similar. And in that area that was interesting, we found my candidate gene, which is, sorry, it's got a horrible name, C17H6 or 52. I promise I will name it properly when I have a mechanism. But it was involved in, it was differentially regulated between the two strains, the fat one and the skinny one. And it just seemed like a really good candidate. So we knocked it out, and so my favorite part about this from a geneticist perspective is that we knocked it out and we allowed cellular repair to just repair it in whatever way it would. So so the Cas9 enzyme did a double strand break, and then the cells were paired one way with a 5 base pair deletion, and then another way with a 10 base pair insertion. So same exact site, targeted, and 2 different repair mechanisms, and yet they have no common phenotypes. So that really tells you that it's a lot more complex than we feared, but like that's still kind of exciting too, because like you wouldn't want to study a complex disease as a geneticist if you weren't if you were afraid of complexity. So that's basically it.
Oh, beautiful. So what is your take-home message?
Rats, it's complex.
And humans as well.
And humans as well.
Congratulations to both of you.
Thank
You
you.
did amazingly well, and you should be really, really proud of the result.
Thank you very much.
Good job.
Thank you.
Yeah.